Hello. In this video, we will be introducing and discussing the different reagents used for cell proliferation, cell viability, and cytotoxicity assays. We will review the relative strengths and weaknesses of each so that you can determine which is best for you in your investigation. We will start with a quick review of cell proliferation, cell viability, and cytotoxicity as concepts before beginning to discuss and compare the most commonly used assay methods and reagents. From there, we can proceed to discuss assays that incorporate measurements of multiple parameters, including measurements related to specific cellular functions. People who are new to these kinds of assays, and even experienced researchers, often feel overwhelmed with the amount of different choices available. Not only are there different parameters to decide to measure, but there are also different assay reagents with different assay principles. We hope to ease this difficulty by organizing the different possible parameters into 3 distinct categories before explaining the principal concept behind each assay and then delving into the strengths and flaws associated with each. As I stated earlier, we can categorize the various methods into 3 major groups. These groups are defined by the specific parameter that the assay measures. First, we have the reagents that measure proliferation, followed by those that measure viability, and those that measure cytotoxicity. Cell proliferation is the capacity of cells to divide and is thus reflected by cell division and DNA synthesis. According to this idea, we can measure proliferation by counting cells or labeling nucleotides. Cell viability is a parameter that refers to the health and productivity of the individual cells. There are a wide variety of viability indicators, often involving intracellular enzymatic activity or products, such as the measurement of esterase activity or ATP or NADH levels. Cell viability measurements serve to indirectly indicate cytotoxicity and the cell's ability to replicate. Viable cells are defined as cells with high enzymatic activity. Cell viability assays are the most widely used category because they often do not require special equipment, and they reflect both proliferative capacity and cytotoxicity. Lastly, we have cytotoxicity, which measures cell death and damage. These methods often measure evidence of cell membrane damage. The reagents used are often impermeable to the membranes of the living cells, serving to measure intracellular substances that have leaked out or by entering and staining dead cells. Within the category of cell proliferation, one method for counting cells is the simple optical method, which can be performed with devices such as the hemocytometer. Alternatively, cell proliferation assays may detect DNA synthesis. There are many methods of measuring cell viability that have been publicized. These viability assays can be further separated into 4 additional categories, again defined by the specific viability parameter that is measured. One category measures intramitochondrial dehydrogenase activity. Another measures intracellular dehydrogenase activity. Another measures ATP levels, and the last measures esterase activity. Let's begin with those that measure mitochondrial dehydrogenase activity. You've probably heard of MTT, which is a reagent from this category that was developed in 1960. MTT is a lipophilic dye capable of penetrating the cell membrane and entering the cell. It then accumulates in the mitochondria and develops color after being reduced by NADH or NADPH, which are products of dehydrogenase activity. The reduced colored form of MTT does not dissolve in water, but forms needle-like crystals. These crystals must be dissolved in organic solvents, and then absorbance can be measured. Next is a category that measures intracellular dehydrogenase activity. Nearly 4 decades after the development of MTT, Dojindo developed WST8 and the associated cell counting kit 8. WST stands for water-soluble tetrazolium. Whereas MTT is lipophilic and capable of entering the cell, WST8 is water-soluble and incapable of entering the cell. Cell counting kit 8 includes an electron mediator, which allows electrons to transfer from NADH inside the cell to WST8 dye outside the cell, causing coloration to develop in the medium outside of the cell. Because the dye is water-soluble, absorbance can be measured directly after adding the reagent and incubating, whereas MTT requires an additional step to dissolve the dye crystals. This water solubility allows for a more quick and simple procedure than the use of MTT. Next, we have the cell viability assays that measure ATP levels. ATP is a very important molecule involved in a wide array of critical cellular processes. Lowered ATP levels are strongly associated with metabolic disorders and processes of programmed cell death like autophagy and apoptosis. One assay in this category involves the use of luciferase to react with ATP and produce luminescence, which can be measured. One large consideration to make with this method is the fact that the cells must be lysed in order for the ATP to be available for detection. Last, we have the methods that measure esterase activity. One example of this method uses a fat-soluble reagent named calcine AM. AM stands for astoxymethyl ester, which is bound to calcine. Calcine AM will permeate the cell membrane before being hydrolyzed by intracellular esterase. This hydrolysis will separate the AM portion, leaving a water-soluble and fluorescent calcine molecule inside the cell. The intensity of this fluorescence is then measured. This technique requires a warning about serum, however. Calcine AM will also be hydrolyzed in the presence of serum, so the medium needs to be changed to serum-free medium prior to the addition of calcine AM and the measurement of fluorescence. Because the inside of the cell is stained, fluorescence can be measured not only by a plate reader, but also via fluorescence imaging or flow cytometry. Now we would like to compare each cell viability assay reagent. An important key is to first exclude reagents that are incompatible with your available detection instruments. Compatible instruments are listed in the first row underneath the name of the reagents. Below that, this table lists the number of publications for each reagent, as well as various advantages, precautions, and recommended usages. MTT, for example, is recommended for cost-conscious users, as it is the least expensive among these options. MTT, however, is not recommended for use with reducing agents due to the fact that they will reduce MTT and produce extra coloration. Cell Counting Kit 8 is recommended for its simple procedure, especially to researchers who are new to these experiments. The simplicity of the procedure will prevent mistakes or errors. Furthermore, cytotoxicity is very low, and samples can be reused after CCK8 assays. Keep in mind, however, that this method is affected by reducing agents as well. ATP measurements of viability are characterized by high sensitivity, allowing them to be used in plates with 384 or more wells. Given that the method is luminescence-based, care must be taken in regards to the half-life of the signal after the reaction. Finally, we have calcine AM and Cell Counting Kit F, the ones that measure esterase activity. Because they stain the insides of cells with fluorescence, they are compatible with fluorescence imaging, flow cytometry, and plate readers. Furthermore, they are unaffected by the presence of reducing agents, unlike MTT and Cell Counting Kit 8. However, it is important to note that depending on the measurement time, there are concerns that reagents may leak out of the cells due to cellular efflux. Now, let's move on to the cytotoxicity category. Similar to the section on viability, we'll identify 4 common methods that detect and measure cell membrane damage. When cell membrane damage occurs, intracellular factors such as enzymes will leak into the medium. Our first category of cytotoxicity assays relies on this. The LDH release assay detects the presence and activity of an enzyme that has leaked into the medium, specifically lactate dehydrogenase, which is a particularly stable enzyme. The assay works by adding water-soluble WST dyes, NAD, and lactate. Lactate acts as a substrate for LDH, and LDH activity causes the WST dyes to produce coloration. In this way, changes in absorbance serve to indicate cell damage. LDH release assays are typically only designed to be used in mediums still containing the sample cells, but Dojindo's LDH assay kit is designed to optionally work in supernatant after the cells have been transferred out. Either way, the procedure is as simple as incubating and then measuring after the reaction has stopped. The next method of cytotoxicity assay is the calcine release assay, which uses the live cell staining dye, calcine AM. The principle behind this assay is similar to the LDH release assay in that we measure the amount of calcine that has leaked out of the cell due to the membrane damage. It is important to note, however, that calcine staining must occur prior to the membrane damage. This method has been used in antibody-dependent cellular cytotoxicity assays, so we will briefly explain that method as well. Target cells are first pre-treated with calcine AM, as staining must occur before any damage. Then antibodies and effector cells are added. Following treatment, we measure the fluorescence of calcine that leaks from damaged target cells. Due to the use of antibodies for targeting, this technique is easy to use in experiments that involve multiple cell types. Our next two methods make use of propydium iodide and trypan blue respectively. Both are commonly used to stain dead cells for observation. Let's start with PI, which is a fluorescent nuclear staining reagent. PI is impermeable to the membranes of living cells. However, when membrane damage occurs, PI will enter the cell and intercalate into the DNA double helix, staining the nucleus as shown in Figure 1. Trypan blue, on the other hand, is a colorimetric probe and is taken up by living cells. However, trypan blue is quickly discharged after being taken up. Although when cell damage occurs, the cell loses its efflux capability, allowing trypan blue to bind to proteins in the cell and turn blue. Similarly to before, we will compare the different cytotoxicity assay reagents. The layout is the same. At the top, we have compatible instruments and an approximate number of publications associated with each reagent. Again, the first priority should be compatibility with the detection instruments you have available. LDH release assay is the most common plate assay based on absorbance, and it is recommended for the dead cell assay and drug screening due to the simplicity of its use and its applicability for screening experiments. It should be noted, however, that it may be affected by the presence of serum, so preliminary experiments should check for any effect by serum. If you have access to fluorescence microscopy or flow cytometers, or if you are trying to co-stain with other fluorescent reagents, consider selecting either calcine AM or propydium iodide, as they are the two fluorometric methods. Calcine release is recommended for experiments that involve more than one type of cell, such as the ADCC assay. Keep in mind, however, that measurement time needs to be tracked and ideally held constant due to the concern of reagent leakage. Propydium iodide, on the other hand, is less expensive and has a very simple procedure. Its emission spectra is often very useful for multiple staining, and leakage is not as large of a concern. Finally, trypan blue is useful for those who do not have access to microplate readers or fluorescence imaging devices. Its signal can be measured with a microscope and hemocytometer. With that concluded, we would like to move on to examples that measure multiple parameters and examples that produce more detailed cell function analysis. In recent years, referees have increased their scrutiny towards measurements of cytotoxicity. There is an increasing demand that researchers prove cytotoxicity by more than a single parameter of measurement, as correlation between multiple factors is more compelling evidence. We will introduce some examples along with a paper. This first example involves a single sample undergoing both a cell viability assay and a cytotoxicity assay. Cell counting kit 8 is being used to measure viability by detecting NADH activity, and LDH assay kit WST is being used to measure cytotoxicity by detecting extracellular LDH. In this experiment, a drug is added to induce cell membrane damage. The graph here indicates the impact of increasing drug concentration upon cell counting kit 8 and LDH assay kit WST results. The x-axis reflects the drug concentration. The blue circles indicate CCK8 signal, and the red squares indicate LDH kit signal. As drug concentration increased, CCK8 absorbance decreased, and LDH absorbance increased. These results respectively indicate that viability has decreased and cell damage increased, which is consistent with the application of a cytotoxic drug. Keep in mind that this combination requires the specific use of Dojindo LDH assay kit WST so that the LDH release assay can be performed on the sample supernatant. Our next example again combines a cell viability assay and a cytotoxicity assay. More specifically, calcine AM is being used to detect esterase activity, and PI is being used to indicate dead cells. This combination is popular in apoptosis and cancer research. When only measuring viability, there are concerns of calcine reagent leakage. Likewise, PI measurements carry the concern of false positives that occur when PI stains RNA in the cytoplasm rather than staining DNA in the nucleus. Using the two together and requiring that their results correlate lowers the fear of these concerns. These are only a few of many possible combinations being used to measure cell proliferation, viability, and cytotoxicity. We encourage you to look for combinations used in your specific field of interest. The assays we have covered so far only confirm the life, productivity, or death of cells without analyzing the underlying elements of the intracellular metabolism. The relationship between intracellular metabolism and cell death has been a growing topic of interest, and we would like to introduce a relevant paper on aging research published in Science this year by Dr. Nakanishi of the University of Tokyo. In his paper, he measures the activity of the glutamine metabolic pathways and conducts cell proliferation and cytotoxicity tests. To the right is a schematic representation of how senescent cells respond to lysosomal membrane damage. Lysosomal membrane damage causes acid to release into the cytoplasm. Cells compensate for the pH decrease by increasing the expression of GLS1. GLS1 is a glutamine metabolizing enzyme that produces ammonia and thereby neutralizes the acid released by lysosomes. Senescent cells rely more strongly on this mechanism and maintain high levels of GLS1. As a consequence, senescent cells produce greater amounts of glutamate and alpha-ketoglutarate than normal cells. Now, understanding the role of GLS1, particularly in senescent cells, they inhibit GLS1, thereby suppressing the glutamine metabolic pathway. This resulted in decreased ammonia production, meaning that acid released from damaged lysosomes would accumulate and ultimately lead to cell death, especially for senescent cells. They then performed cytotoxicity tests on both senescent and non-senescent cells, using calcine to detect live cells and LDH assay to detect dead cells. These tests confirm that GLS1 inhibition was much more deadly to the senescent cells than to the non-senescent cells. By analyzing both the intracellular metabolic pathway and different parameters of cytotoxicity, the researchers were able to link cytotoxicity and cell death to a particular cellular metabolic pathway. Compelling results like these contribute greatly to our understanding of disease mechanisms and more convincingly highlight drug discovery targets. This concludes our experiment examples, although many more possibilities exist. If you are interested in any of the particular metabolic indicators in the diagram to the left, please refer to our product lineup. If you are interested in learning more about Dojindo's cell proliferation and cytotoxicity reagents, our experimental protocols are available online, including guides on troubleshooting. Additionally, please feel free to contact the Dojindo representatives in your region. And finally, here we have a list of Dojindo products that correspond to each of the different ideas introduced today. This list, however, excludes the various reagents we produce to detect elements of the intracellular metabolism. If you are interested in those, please visit our website and check out our Metabolism Map, a guide to cellular metabolism. We have reached the end of our presentation, although we have much more information to share. Please feel free to contact us for any additional questions you may have. Thank you very much for your attention.